Happy Monday, everybody. I hope you're having a good day, whether it's morning or afternoon when you happen to see these notes. So these are what you're going to do today while I am not in the office. It is currently Sunday when I'm recording these. I've had a fun-filled day of playing with the pup, trying to tire her out before her big procedure tomorrow, cleaning, meal prepping, grading, all the fun things that accompany adult life. So whenever you're watching these, I hope you're having a good day, and these are going to be used to accompany your fill-in notes for Chapter 9, which is all about cell respiration and fermentation, as you can see here. Make sure you've done your bell work. There is a bell work posted for the 12/7 presentation. There's a question there in an I can. Make sure that gets handled. And again, like I mentioned on Friday, if you didn't happen to hear it or catch it, you must be filling these assignments out, bell work, and these notes in order to be considered present today, seeing that you do not have a substitute filling in for me. So let's get started. We talked a little bit on Friday about cell respiration and fermentation with our crash course notes, and you've dipped your toe into the aerobic respiration deep end, so to speak, with your TRG. So let's kind of tie everything together and end Unit 3 on a high note in terms of cellular energy. I've got my next slide. There we go. All right, so these are going to focus on respiration, okay, taking in those materials that you need, oxygen, and eating that other reactant that we're going to need to run this process, glucose. Just for quick reference, this is going to be done by anything that has a mitochondria, so this is going to be terrestrial organisms, as well as your photosynthetic organisms that dwell not only on the land, but in the water as well. Just to take a quick look at the equation itself, the molecules that are going to be oxidized in this reaction are glucose. Oxidation, again, we're giving away electrons, generally breaking bonds. It makes sense that we're breaking down that food particle into a smaller molecule, which is going to be CO2. What's going to be reduced and what's going to be our final electron acceptor, the most important molecule in terms of evolution in this process of being able to use oxygen to run respiration, is oxygen. So oxygen is going to be reduced by becoming or combining with hydrogen to form H2O. And you can see here, as glucose is broken down, where did all those hydrogens go? Well, they're going to be shared between our electron carriers and water molecules. Okay, ultimately doing these two processes to yield energy. Wrong way, sorry. You may have noticed on Friday when we talked about this process, we had an electron carrier that was a little new to us, NAD or NADH, in comparison to NADPH, which you saw in photosynthesis. And you can see here, the molecules look very, very similar. However, this is accompanied by a phosphate molecule. So this may be a component that is important for that specific electron transport chain that was not present in what we're going to go through today in terms of the electron transport chain found in mitochondria. But in terms of their function, very, very similar structure matching function, except for this one chemical difference. We're also going to talk about the electron carrier FADH2 or your FADs. Okay, so this electron carrier is going to be a little bit bigger in terms of structure than NADH or NAD. Okay, but the function is going to be the same, carry electrons from splitting those molecules to the final step of the process itself, generating ATP. Okay, so here's an overview. Let me move myself a little bit of the whole process. We learned a little bit about glycolysis, glycolysis, taking glucose, that's 6 carbon sugar, and splitting it, 6 carbon sugar and splitting it into 2 molecules. And you're going to notice that digestion of that molecule is going to occur over several steps outside and within the mitochondrial membrane. So that first breakdown is going to create pyruvate molecules. Okay, that breaking of those bonds is going to generate a little bit of ATP. We talked a little bit about fermentation and how this process here for organisms that do not have a mitochondria or do not have access to oxygen are going to rely upon this process. Multicellular organisms like ourselves and others are going to use that addition of oxygen to run this electron transport chain and oxidative phosphorylation. I know, scary word, but that just means generating ATP using oxygen. This diagram here has the intermediate step or the preparatory step of making this coenzyme that's going to be used later on in the process itself. So that is taking pyruvate and breaking it down into this form. The citric acid cycle, we learned that is also known as the Krebs cycle. I'm going to refer to it as the Krebs cycle because that's like what I like to call it. But we're going to get rid of CO2 in this step, and we're also going to generate our electron carriers and get them charged up to finally deliver electrons and create a chemiosmotic difference to pump out some ATP here. And you can see across the steps, we're generating a little bit of ATP here in glycolysis, a little bit in Krebs cycle, the majority in the electron transport chain, with the addition or having access to oxygen. So in Hank's video on Friday, if you remember back, we spoke a little bit about fermentation. So for organisms that do not have a mitochondria, aka prokaryotes, this is a process they rely upon. Through evolutionary connection, this is a process that occurs among all living organisms. If it's considered a living organism, it performs glycolysis. So that shows that there is commonality among all forms of life in terms of this energy generation. So in glycolysis, again, we remember we're breaking down glucose into pyruvate if we do not have access to oxygen. So including organisms such as ourselves who have a mitochondria, but in times when oxygen levels are low or it's not being processed quickly enough, fermentation is going to occur. So we are going to perform lactic acid fermentation. This molecule itself, pyruvate, is not going to move into the mitochondrial membrane. It's going to remain within the cytoplasm. It's going to be broken down into lactic acid, and only over time is a cell going to be able to break down that lactic acid and get it out of the cell to change that acidic condition within the cell. If we're talking about bacteria or some plants, they're going to be able to do fermentation and create ethyl alcohol, which we utilize for many fun things, from baking to dairy production to wine, spirits, beer. And one byproduct of that process is CO2. So you can think about that if you've ever made bread and you've used the active yeast, you put the yeast, warm it up, wake it up, so to speak, and you let that material rest. If you've added, flour and baking soda and salt to it, it's going to rise. And what's rising or causing the rise within it are those CO2 bubbles as the yeast itself go through this process of fermentation. So they're going to have some access to glucose there. They're going to break it down into pyruvate. And within that bread mixture, some ethyl alcohol, which cooks off, you're not getting drunk off of bread, is going to be created, and the fluffiness of those products is going to be due to CO2. Now, if we have access to oxygen, this intermediate form, this coenzyme is going to be created. That's going to move into the Krebs cycle and then eventually be used to generate energy carriers and finally be delivered to the electron transport chain, ultimately getting some ATP, We're spending money to make money. So you can see that these are our money steps that happen along the way. And this is our input, glucose, eventually yielding all of this ATP. If we're talking about fermentation, you may hear it referred to as anaerobic respiration. Anaerobic means without oxygen. It's not going to require oxygen. And we mentioned that could fall under two umbrellas, alcoholic fermentation or lactic acid fermentation. I'm going to discuss, move myself a little bit more, a few types of phosphorylation throughout this process. So phosphorylation, just a fancy term for adding a phosphate group. This could happen at the substrate level. This is essentially just a handoff. One molecule is giving another molecule phosphate. So for example, if we're talking about phosphorylation that occurs in glycolysis, which we're going to look at in a second, that would be considered substrate level phosphorylation. If we're talking about oxidative phosphorylation, this is a phosphorylation that occurs during the electron transport chain. Okay, it's going to require oxygen. So as ATP synthase, okay, puts together ADP and that phosphate, okay, it's going to be using oxygen to help that process occur. And you'll see in detail today how we use that oxygen molecule to keep this process pumping. And you could probably figure out just through etymology what this last one is, photophosphorylation. We're probably using light here, okay? So this is going to be associated with light and how light helped to move those electrons down the electron transport chain in photosynthesis. So that light was the catalyst that was helping to move those electrons down those proteins to eventually get to ATP synthase to make some ATP for plants within the chloroplast. Okay, we are using oxygen, anything that has a mitochondria, us, plants, fungus, and so on. We're using that oxygen to generate the ATP in the electron transport chains through oxidative phosphorylation. Okay, so first step, glycolysis. Taking glucose and breaking it in half, 6 carbon into 3 and 3 to generate pyruvate. Okay, what are we getting from that breakdown? Well, we're getting some electrons released, and that's going to be carried by NADH. And you can see NADH is going to carry that electron all the way to the electron transport chain. Okay, we're also putting in 2 ATP to get this party started. You're going to use 2, okay, but we're going to yield 4, meaning that we got a total of 2 output. So you got 2 here. Since 4 were generated, but we used 2, we got a net of 2. So what I want to do really quickly, let's pause here and we'll go to our fill-ins. Okay, let me get out of this. So let's go here. I'll zoom in just a tiny bit and kind of read these out to you guys. All right, so under basics, okay. So for cell respiration, we're breaking down carbs to build biomolecules. We're consuming glucose, and we're producing ATP and not oxygen, water. Sorry for that one. Produces ATP and water. The equation can be seen below. In terms of energy, our high energy molecule here, which we are breaking those bonds to get those electrons from, is glucose. Our low energy molecules, which are smaller, okay, in comparison, C6H12O6 has a lot more bonds to break than CO2 and H2O. Our electron pathway, it's aerobic, so we're using oxygen. Glucose is going to get oxidized. We're going to steal electrons from it, remove that negativity. Okay, and those electrons are going to be passed to electron carriers. Our electron carriers are our NADs and our FADs, our NADHs and our FADH2s. Okay, the reaction of cell respiration allows energy and glucose to be released slowly. Therefore, ATP is produced gradually. Still, it's only 39% efficient, meaning that all the energy that's available within that glucose molecule, you're only getting 39% out, okay? A lot of it's going to be lost as heat. We're going to have that heat conversion. Our coenzymes, which we are going to talk repeatedly about today, are NAD and FADH2. They're not going to be consumed, just as it wasn't consumed, NADPH, in photosynthesis. They're going to be recycled. Okay, here's our steps. We talked briefly about them. If they're aerobic, meaning that they have oxygen, and if they are anaerobic, meaning no oxygen, that would be fermentation in its 2 forms, alcoholic and lactic acid. Okay, and then in terms of our phosphorylation types, we discussed substrate level phosphorylation, which is passing the phosphate group, one molecule to another, just pass it on. Oxidative, which requires oxygen, and that's going to be the final electron acceptor at the bottom of the electron transport chain in the mitochondria. Okay, this is a big finale, but this is what allows the electron transport chain to keep pumping out more and more and more ATP. And then in terms of photophosphorylation, we think back to photosynthesis, which is associated with light in the electron transport chain of the chloroplast. Okay. So we talked a little bit already about about glycolysis, so let's fill in that one under overview. Okay, it's going to occur in the cytoplasm. By prokaryotic, we're going to also add anaerobic cells. Okay, so cells that do not rely upon oxygen. It's going to break down glucose. Okay, if you decide to go to med school, you're going to have to look at and learn all 10 steps of glycolysis. It doesn't seem so in-depth to take a 6-carbon molecule and break it in half, but it is. So have fun with that if that's a path you choose in life. And what's important in terms of evolutionary relatedness, it's a universal reaction. Okay, it's probably going to be evolving first before other types of energy generation or energy transferring among organisms. What's our energy investment? We're going to net 2 molecules of glucose using 2 ATP. Okay. And then glucose is going to be split into that 3-carbon molecule called, we're missing it here, pyruvate. Called pyruvate. All right. So let's skip on down to the basics here in terms of fermentation. So we talked about it being anaerobic. Okay, again, it's going to be lactic acid fermentation or alcoholic fermentation depending upon the organism. So our basics is going to be organism here. What's the purpose of it? The purpose is to oxidize NADH so that it can be reused in glycolysis again. Okay, so you're going to generate that NADH, give an electron from glucose to that molecule so it can be used and spent and then returned to the process to do it again. Some advantages of fermentation. It's a quick burst of energy for organisms. So if you are a sprinter, if you've had to sprint at any point in time, the electron transport chain or cell respiration in general takes a while to warm up. There's a few steps for it to go through. Glycolysis is quick. So if there's a quick need for energy, your muscles are going to utilize the energy that's already stored there in your mitochondria, and then it's going to perform lactic acid fermentation to get a quick 2 ATP, okay, while it's waiting for the energy to be generated through the electron transport chain for cell respiration. I didn't mention it yet, but you can use this process, lactic acid fermentation, in bacteria for dairy production. So certain dairy products like yogurt, sour cream, things that have a little bit of a sourness to them, those bacteria are going to go through the fermentation process and it's going to impact the flavor of the dairy product. So more soury or savory dairy products, they're going to go through more lactic acid fermentation in comparison to those that are going to be a little more sweet, like ice cream and so on. In terms of disadvantages, we're only making 2 ATP from glycolysis. In terms of lactic acid fermentation, you're going to have a huge oxygen debt. Okay, you're going to be breathing heavily, and it's required for a certain amount of O2 to re-enter the cells and rid those cells of the lactate. Lactate is an acid. Okay, it's going to be toxic to the cell. It's going to change the pH, and it's going to need to leave the cell in order for that pH to re-regulate and the cell to go back to normal functioning. So there's obviously a disadvantage to that, and you can feel it. You feel that your body's out of whack when there's a lactic acid buildup. Okay, and in terms of efficiency, when we perform cell respiration, we're only getting 39% of the available energy from glucose. Look at fermentation, only 2.1% of the potential energy of glucose. So very inefficient, quick energy in terms of 2 ATP, but very inefficient in the breakdown of glucose. Okay, so let's go back to our notes. right? So here's just a very simplified pathway for glycolysis. You can see we have our glucose molecule, C6H12O6, breaking down into pyruvic acid. We see the carbons are in half, the oxygens are in half, but that's not half for hydrogen. Where did a few of those hydrogens go? Well, they were used to reduce or give those electrons to NADH. Okay, so we went up from NAD plus to NADH. Okay, they're going to be left there and held by that little electron carrier donkey and be delivered to the final step. Okay, this would just be the example of substrate level phosphorylation. So the enzyme itself is delivering or providing this phosphate molecule to ATP in order to give it its shape, or its 3 phosphate molecules here, went from ADP to ATP. So that handoff from the enzyme here to the substrate, ATP, that's substrate level phosphorylation. It's just a handoff. All right, so we talked about that regularly. Let's go past. Okay, a little bit more about anaerobic versus aerobic. If you need another diagram to look at, there's fermentation again, which we've discussed repeatedly. So we're just going to continue on as I move myself around, sorry. Okay, so in this diagram itself, we've talked about glycolysis happening in the cytoplasm or the cytosol. We have some substrate level phosphorylation, okay, where we're handing off a phosphate to generate some ATP. We've got 2 of them. If oxygen's available, we're now going to head into the mitochondrial matrix to perform our Krebs cycle and then eventually our electron transport chain. And you can see electrons are being carried from glycolysis to electron transport chain. And in Krebs, we're helping to generate more electron carriers to deliver electrons to the electron transport chain. In Krebs, remember, a main part of that is getting rid of carbon dioxide that's toxic to our cells. And then you're going to find out where we're generating or reforming these water molecules has to do with oxygen. Okay, so here is the anatomy of the mitochondria. This is an electron microscope picture here. You can see that it has that double membrane. There's a lot of surface area to volume ratio happening with all the folds. The inner space is a matrix. This is where the citric acid cycle or Krebs cycle is happening. And then in the cristae, which you can follow here, so that cristae is like inside of these little pathways. That's where electron transport is going to happen. Okay, it needs a membrane, so there's a membrane for it to occur upon. Remember back to the thylakoids also had a membrane that it would work through, the electron transport chain. Same idea for the mitochondria. All right, so again, here's glucose being broken down into pyruvic acid. That's what's going to enter the Krebs cycle. Okay. So here's what's happening during the transition reaction before we get into the Krebs cycle. Okay, we're going to make a molecule that's going to help enzymes later on in this specific electron transport chain. Here's the chemical structure for pyruvate. We see it's a 3-carbon molecule. Makes sense. Glucose is 6. We split it in half. And what we're doing, we're removing this COOH, okay, that carboxyl group. CO and O are going to leave as carbon dioxide, right? CO2. And then this H is going to be given to NAD to reduce it to NADH. So we have an energy carrier, we have a byproduct to get rid of, and now we have a coenzyme that's going to enter the next step to help perform the process of forming ATP. Okay, so we went from pyruvate to acetyl-CoA. We got some CO2, which we're going to get rid of, and we have a little bit of an electron carrier, NADH. So we get one from this part. We also got one from glycolysis. We're going to get even more during the Krebs cycle. Okay. So Krebs cycle looks crazy, but here's all you need to know. Okay. Carbon dioxide is leaving. That's important. That's a byproduct. We're also taking our input molecule, which is acetyl-CoA, and we're changing up its structure. And as we change up its structure, going from a 6-carbon structure to a 5-carbon to a 4-carbon, okay, we're getting different electrons as we break those bonds. So it's going to combine with a few other molecules to make a 6-carbon molecule. And then it's going to be broken down in a few steps. And as it's broken down, we break bonds, we get energy. And what are we doing with that energy? We're putting it onto those electron-carrying little donkeys and prepping them for the final step, okay? So we're breaking bonds to collect electrons and carry them to the next step. We're also getting rid of carbon dioxide, okay? So that's the main 2 ideas under the Krebs cycle. Break some bonds between a carbon molecule, collect those electrons, and get rid of CO2. Okay? You're doing a good job. Stay with me. It's not as difficult as it looks. All right, so here is what our inputs were and what our outputs are for Krebs cycle, okay? So we have 2 acetyl COAs, we have some NADs and some FADs, and we had a little bit of ATP. What are we getting out? Here's our charged up electron carriers, our NADs and our FADs. There's our CO2 we're getting rid of, and we generated just 2 ATP molecules. Okay, so we got a little bit of energy. Our electron carriers are locked and loaded. They're ready to roll, and we've gotten rid of our byproduct. Okay, so before we get into electron transport chain, let's get out of here. Let's go back to our notes. Move me over here. Okay. All right, so the prep reaction, that was going from pyruvate to acetyl-CoA. It's going to connect glycolysis to the citric acid cycle. It's connecting that molecule we started with the molecules that we got out of the Krebs cycle. When I say citric acid, same thing as Krebs, okay? It's like saying Calvin and light independent. They're going to be synonymous with one another. All right, we talked about removing that carboxyl group. We got some CO2, and then we're using those hydrogens to get our electron carriers prepped. Okay, so Krebs cycle, it's going to complete the oxidation of the original glucose. Oxidation means you're removing electrons, right? We took some electrons from it, we stole it, we're using those electrons to prep our electron carriers. Oh, goodness. Alexa thinks I'm talking to her. Hold on. I apologize for that. I don't know how any of that was associated with Alexa. All right, so from our glucose molecule, the cycle occurs 2 times. Okay, there is a song that goes along with this. We're not going to worry about it. Okay, we move some molecules around. We build up a carbon molecule. We break it down. As we break it down, we collect those electrons to give to our energy carriers, which would be reducing FAD and NAD. All right, so we're at electron transport chain, taking all of those electron carriers to eventually get ourselves a lot of ATP, because thus far we've only net 4. We got 2 from glycolysis, we got 2 from Krebs cycle. That's where we're at right now. We need a whole lot more. Okay. All right, so here's the electron transport chain. I think I like this picture a little bit better. So here are the electron carriers, carrying electrons from food, because essentially we're breaking down glucose, or that's what we've done. Here's NADH, here's FADH2, dropping off electrons. So those electrons are going to make their way to each protein in the membrane, and if you notice, as they touch or pass through each protein, you can see a hydrogen proton is pumped out, okay, from inside out. Different than photosynthesis. Photosynthesis electron transport was out in, but we're building up that chemiosmotic difference. A lot of pluses here, not so many there, because we know when we have a concentration difference, molecules are going to move. So as the electrons move through each protein, so it passed through this one, hydrogen moved out. Move to this one, passed it over here. This electron caused the hydrogen to pump out. Then it moves over here. This electron caused the hydrogen to move out. And those hydrogen protons build up in the outer space of the mitochondria, and they're going to cause ATP synthase because they're going to passively move through this enzyme to cause it to turn. Remember, it kind of functions like a gumball machine. You put a quarter in, it twists. It literally physically twists in the membrane and puts together ADP to ATP. Okay? Now, what's going to happen over time is those hydrogens are going to move their way through ATP synthase, and that concentration gradient is going to increase down here inside the mitochondria. Well, we're going to be deadlocked here unless we clear those hydrogens out. Okay, you want to keep this space clear so the electrons can keep moving through. So NADH and FADH2, our NADs and FADs, can keep dropping off electrons. If there's not a concentration gradient, nothing's happening. Okay? So who's going to take care of those hydrogens? Well, that's where oxygen comes into play. Oxygen is going to couple with those hydrogens and form water, okay? And that's going to remove those hydrogens, keep that concentration difference, and keep ATP synthase pumping as long as there's electrons to move hydrogen protons through. Okay, so that's the name of the game. Electrons change the gradient. Once the gradient is changed, ATP synthase is going to turn. And as long as there's a difference, it's going to keep on turning. All right, so here's another version of it. Okay, so you can see glucose here. There's our net of 2, pyruvate. Here's your transition reaction from pyruvate to acetyl-CoA. Goodbye, CO2. What are we getting out of that? NADH here in glycolysis, NADH from a prep reaction, NADH and FADH2 from the Krebs cycle. Where are they all going? To the electron transport chain. Why? To get all of this ATP, okay? And then what's taking care of those hydrogen protons again? That was oxygen forming water, okay? Here's another picture. I think I like this one the best. Showing the process all over again. I like this one because it shows if oxygen is present. So you can do oxidative phosphorylation using oxygen to generate ATP. This side is without oxygen. And you can see glycolysis without oxygen, you do reduce NADH to be used for some fermentation. Okay, in terms of our cells, that would make lactate, and that's what eventually has to leave the cell. But when oxygen is present, we take glucose, break it up, okay, prep it to acetyl-CoA, go into the Krebs cycle, and then get those electron carriers over to electron transport chain and ATP eventually out. All right, and then here's another version of it, if you'd like to look at it a few different ways. There's something in this picture I want to mention, though, in terms of what molecule you can use at what part in this process, because surprisingly enough, it's not just a carbohydrate, okay? You can use fats and you can use proteins in this process. All right, so let's go back to our notes really quick. We're on electron transport chain. Myself over here. Purpose of electron transport chain, to reduce our NADs and our FADs, which bring electrons to the ETC. They become oxidized, meaning they have given their electrons away, okay? And they return to the other steps of the cell respiration process. Okay, as electrons pass down the ETC, their energy is captured to pump hydrogen ions in the intermembrane space. Once they're built up, we have that chemiosmotic difference. They go through the ATP synthase and get ATP. Okay. Few things about anatomy and ATP production. We've already discussed those. And then most importantly, because we are doing oxidative phosphorylation, oxygen is going to bind to hydrogen to form water. Okay, and that's how we can keep that process going. And that's how we get so much ATP from that final step. Okay, in terms of energy yield, here's where we're at. 2 from glycolysis, 2 from Krebs cycle. Okay, these specific molecules themselves, our energy carriers, are going to be responsible for the majority of the ATP collection or generation. And then again, efficiency-wise, from our original glucose molecule, not very good. Okay, we're losing a lot of energy or transferring a lot of energy to the form of thermal energy, heat, as this process occurs. So the numbers below are older. New evidence in terms of the whole process of aerobic cell respiration is that more like 30 to 32 total occur from the process itself. Okay, before when I learned it, and a lot of diagrams still show about 38. Okay, so it's going to be in the realm of 30, depending upon metabolism itself and the access to oxygen and glucose. Okay, so let's go back here. Two more things. You're doing good. Stay with me. I know it's a lot, but you can pause it and take a break as needed. Okay, so just a quick attention to catabolic and anabolic pathways. So catabolic, we're breaking down food molecules. Those could be glucose in the form of carbs. Could be fats. We'll talk about how fats are going to be metabolized. Could be proteins. And we're going to turn those into usable forms of energy to eventually build up other molecules. Okay, so what other molecules our body is going to use that energy for depends upon what processes need the energy itself. Okay, so moving material, fighting infection, building other biomolecules, locomotion in general. Okay, and then with all of those catabolic pathways, that energy is not going to be a complete transfer. Some of it's going to be lost as heat. Okay, so this is where the other biomolecules are going to come into play. Let me make this up a little bit bigger. So we know carbs. Carbs are going to go in as glucose. Glucose is going to be broken down in pyruvate and so on. Proteins, okay, in this picture up here, this is a pan with like a stir fry or something. So within this pan, we're going to have proteins, carbs, and fats. Proteins can enter at 3 different locations in aerobic respiration. They can enter as amino acids. They're going to be broken down to their building blocks. Those amino acids can enter glycolysis and be broken down. They can enter and be converted into acetyl-CoA, or they can enter the Krebs cycle as one of those carbon molecules that we're going to eventually rearrange and collect some electrons from. Fats, on the other hand, fats can be broken down into glycerol, that's the head of a fat, right? And their tails, the fatty acids. Glycerol can enter glycolysis and be broken down into pyruvate, whereas fatty acids can be used to generate acetyl-CoA. Okay, so that's nice in terms of our fitness, because if you don't have access to glucose, there are other molecules in the body that can be metabolized to run cell respiration. Okay, our cells are so well adapted for this process. It's crazy when you think about it. All right, so just a side-by-side comparison, because this is a big component of this unit, seeing the commonalities and differences between these two organelles. So chloroplasts, God, if you walk out of my classroom and you don't know that they're just in plants and photosynthetic bacteria, I'll cry. Mitochondria are going to be in both. Mitochondria are in both, okay? Plants also have mitochondria. Chloroplast is going to be anabolic because we're storing energy. Mitochondria are going to release it or be a catabolic reaction. Okay. You're going to need to absorb sunlight in chloroplast. Again, you have to have chlorophyll, those light-absorbing pigments. Mitochondria does not rely upon sunlight because they do not have chlorophyll. Okay. And again, we know structurally they're very similar in terms of their use of surface area to volume. Okay, and this picture is so great. I know it looks busy, but it really does show you how the steps of each process occurs in terms of use of electrons and the chemiosmotic differences. So this is what we just went over for cell respiration. See all those hydrogens outside the cell? Okay, those are what are pumped out by your electron carriers, and then they get to usher their way in through the ATP synthase, okay? And eventually oxygen is going to be the final electron acceptor and form water. But that gradient that we're building up is outside of the central space. Whereas in chloroplast, we were pumping hydrogens into the chloroplast, okay? So ATP synthase was taking those hydrogens and pumping them out, okay, to form ATP out of the membrane. So same processes, just how they use that chemiosmotic difference is just opposites. This one, we're pumping hydrogens into the space. This one, we're pumping hydrogens out, okay? But same type of components. Okay, and then lastly, in terms of Unit 3 and what you need to know, it's important to note cellular fitness, meaning that there are a large variety of molecules that we're not only using for metabolism, but that we're also going to, once metabolism is done, form with those products or with that ATP. What are we using that ATP for? Okay. So just a quick recap. So we can use our carbohydrates, we can use our fatty acids, we can use our amino acids to run our metabolism. What are we getting from them? Well, with amino acids, we're getting as many types of proteins as you possibly can, okay? That's high fitness for us because there's such a variety of molecules we can use in terms of proteins throughout our bodies. Fatty acids are going to build phospholipids. We've seen now phospholipids are everywhere. Okay, let's go back a couple slides and look at these membranes. There they are. They're everywhere. They're around every organelle. They're within every membrane that we see in the cells. Okay, so those phospholipids are very important in terms of cell usage and cell fitness. Carbs, again, we can store them as glycerol. We are going to break them down into monosaccharides. We can store them as polysaccharides. We can use monosaccharides to build things like nucleotides, and then our nucleotides are going to be used to form our genetic molecules, which are DNA and RNA, which then code for cell processes to do all of this all over again. How interconnected is everything? Okay, so that is it. I've said a lot of words. I've spit a lot of science. So let's go back to our key sheet and we'll finish that up, okay? Good job. Thank you for following along with me. I hope my obnoxious voice didn't annoy you too much. Okay, so quick recap of catabolism. So we talked about cell respiration. Yes, we're using glucose to break it down to eventually yield ATP, but proteins can be used in all three steps as well as fats. So A is protein, B is fats. Okay. When we had our side-by-side for similarities, we talked about the use of the membranes and the electron transport chain. In terms of flow of energy, we're going from the sun to our chloroplasts, and chloroplasts are creating products through photosynthesis that will be used in the mitochondria for cell respiration. All right. And then finally, under fitness, one says naturally occurring diversity among and between components within biological systems affects interactions with the environment. So it's important to note all of the diversity that not only exists within populations, but at the cellular molecular level. Okay, so A, at the cellular level, it gives organisms the ability to respond to a variety of environmental stimuli. So if we think about cell respiration, there's a variety of molecules we can use to break those bonds and transfer that energy to ATP. So under B, there's a number and type of molecules within cells that provides organisms with a greater ability to survive and reproduce in different environments. And we're done. Yay. Okay, so that is cell respiration. Tomorrow we're going to work through some diagrams and do some labeling and some notes together. So if you do have access to coloring apparatuses, I think that's right, plural, make sure you have them or get them out and have them ready for your Tuesday class. I'll give you guys a little update on Little Miss Penny on Tuesday, and I hope you have a wonderful day. Thank you for paying attention to these notes and filling these in. And I hope you have a great day, and I'll see you on Tuesday. Bye, guys.